Great. Well, now I'm really pleased to introduce Srini Turaga, our second speaker. Srini is a group leader at the HHMI Genelia Research Campus and is also a member of the NSF Institute for Artificial and Natural Intelligence. Srini reminded me that we were actually on the same hallway for a brief time. He was a PhD student at MIT with Sebastian Sung, and he was part of the very early wave of scientists focusing on AI-based approaches for reconstruct the neural connectome. So the synapse scale wiring diagram of the central nervous system, and was part of the iWire consortium that focused on reconstructing the wiring diagram of the mouse retina. He was then a postdoc at the Gatsby Institute at University College London, where he focused on computational approaches for interpreting neural activity. And now in his own group, he's bringing these two approaches together, using AI approaches for connectomics, for reconstructing the structure of the nervous system, and also approaches for measuring and understanding the activity of the nervous system. So please come on up. Thank you.
Thanks for, excuse me, thanks for having me. So I'd like to tell you about work that we've been doing in our lab over the last 4 or 5 years on building simulations of the fruit fly, its nervous system and its body. And this is work done in collaboration with lots of colleagues at Genelia, folks at Google DeepMind, as well as a close collaborator at University of Tübingen, Jakob Maake. So the fruit fly, which we study, the reason we study it is it's a tiny insect, about 2.5 millimeters big. Its entire brain is the size of a poppy seed, which is small. And with this tiny brain, it can do quite a bit. You know, it can crawl on surfaces, it can fly, it can court mates, it can escape predators. It's a highly visual animal, so it does quite a bit of visual processing as well as skillful motor control. Now, something that's super exciting that happened just a couple of weeks ago is that we now have a connectome for the entire nervous system of a male fruit fly, male adult fruit fly. And so we now know that this entire nervous system has 166,691 neurons. That's exact, that's how many neurons it has. And we know exactly how it's wired to the other neurons within that nervous system. So this is something that my colleagues, not from my lab, but my colleagues at Genelia put together and released just a few weeks ago. But a similarly exciting milestone was achieved 2 years ago, where colleagues at Genelia, as well as Princeton and Cambridge, put together a connectome for just the brain part, in this case, a female brain. And this visualization shows you, each color here is a different neuron, and it's quite densely packed inside the brain. But when you look at subsets of these neurons, for instance, descending neurons are neurons that go from the brain to the body. Motor neurons are neurons that directly control muscles. So when you look at subsets of them, you can see the beautiful structure. These sensory neurons, there's the photoreceptors, for instance, as well as many others. The beautiful shapes, as well as the details of exactly how they're all wired up and connected to each other. So in my lab, we're trying to figure out how do we take this structural information about the brain that comes from a dead brain and use that to inform how the living brain computes. And so here's my caricature of how the brain works as a computational neuroscientist. So at this bottom, at the microscopic scale, are the mechanisms of how the brain works. So this includes the cells, the brain cells, neurons that comprise the nervous system, as well as the molecules that are proteins that are made by these cells that control biophysics of neurons, the biophysics of synapses. Now, very excitingly, at this microscopic scale, we now have direct measurements of the connectivity, but we don't have measurements of the neuronal biophysics or synaptic biophysics at this microscopic scale comprehensively for every single neuron. If we knew all that, if we had perfect measurements of everything at the scale, then in principle, we can simulate the collective neural activity that's produced by all of these microscopic components, the neural activity across the entire brain. And if we had neural activity across the entire brain, then we could predict the behavior produced by the animal. But we do not have complete measurements of everything at this microscopic scale. These measurements can only be made in a living brain if you want to try to understand the dynamics of how living brain cells and synapses work. Whereas, this is the only thing you can get from a dead brain. So for this reason, about 20 years ago, when people started talking about, let's use 3D electron microscopy to build these maps, these connectomes, there was a great deal of pushback. And this was literally debated. And this is a debate from about 13 years ago. There's a debate between Sebastian Sung and Tony Malshan, famous scientists, moderated by NPR scientific journalists. And the debate was really about if you can only get this incomplete information at the microscopic scale, is that enough? And it's clearly not sufficient to explain everything. And so why spend millions of dollars to do this? Thankfully, the connectome projects continued regardless and provided the connectomes, which I will now tell you about. And so in my lab, we asked the question, even though we can only measure the connectivity comprehensively, there's plenty of measurements that we can make at these other scales. We can measure behavior that animals produce. We've been able to do that with our eyes and now with high-speed video cameras for ages. We can measure neural activity sparsely, and we can measure a bunch of these things sparsely, not comprehensively, but we know something about what's possible. And so the question we asked is, can we use these measurements that we made, that we can make at the macroscopic scale, and use them along with AI methods to constrain these parameters that we cannot measure at the microscopic scale? And if we can do that sufficiently well, then maybe we can still bring these incomplete microscopic measurements to build a model of the whole brain. So in particular, I'll tell you about a study where we looked at a very extreme version of this. Let's pretend that we only have the connectivity and we only know fly behavior. Can we predict neural activity without ever having provided it to the model? So can we predict neural activity in the living brain using measurements, only measurements from a dead brain? So the idea was to take these connectomic measurements from a fruit fly, in particular the fly visual system, build a model, and see how well we predict the neural activity in the living brain. So quick introduction to the fly nervous system. The fly is a highly visual animal. It has two very large compound eyes that dominate its head. Similarly, the nervous system of the fly is dominated by the optic lobes, which are the visual processing centers of the nervous system. It has a highly crystalline hexagonal lattice organization to its photoreceptors, the pixels. And similarly, there's a highly crystalline hexagonal lattice organization to the nervous system that process these visual inputs. And they come in a sort of layered architecture that we've known for quite some time. What's now exciting is that we had the connectome. This was before the whole brain connectomes were published, so we only had the connectivity for part of it, but it's still quite comprehensive. This is about 64 cell types that take you all the way from the retina to the end of the optic lobe, which are the lobule and lobule plate. What you see here is a matrix where each row is a cell type and each column is a cell type. And there's a square if those two cell types are connected. We know through other measurements of protein expression, we can figure out which neurons are excitatory and which ones are inhibitory. Neurons typically have only one sign to their output synapses, and we know what those are. And the sizes of these boxes tell you how strongly they're connected. And this is something you can get from the connectome. You can just look at these electron microscopic images and these reconstructions and count how many synapses there are between any pair of neurons. And it can range from a handful to 100 or several hundred connections between any two neurons. And so this, we think, is a proxy for strength. Now, we don't just know the connectivity at the level of cell types. We actually have extremely detailed information at the level of individual neurons. And so at this point, we appeal to convolutional neural networks. And you can kind of think of each of these cell types as a feature map. And what we know is actually the filter that takes you from one feature map to another. So each of these squares really corresponds to a filter for which we have really detailed information. And so that's the level at which we have from the connectome. So this allows us to build a simulation that kind of looks like this. As I said, each of these hexagons over here is a feature map, basically the population of neurons of a particular cell type. Each single pixel inside of that is one neuron of that cell type. Here are the photoreceptors. There's 8 types of photoreceptors, 8 of them, which then propagate their activity down into the lamina, the medulla, and so on. The dream, what we'd like to be able to do is, given a particular light pattern that we display to the retina, we would like to be able to predict the neural activity across the rest of the nervous system. And so this is a really good match to the real system in that each of these neurons corresponds to a real neuron in the brain, and each connection corresponds to a real connection that exists in the brain. So that's the level of detail that we can now build this model. Now, if we want to build this and sort of bring this to life, we need equations. So we have to give each neuron and each synapse equations, ordinary differential equations. And we don't know what those parameters are. If we knew them, then we could simulate them. We don't know what those are. So we have to figure out how to fill those in. Now, even, don't stare at this too long, but even the simplest approximation you could build for how a single neuron works or how a single synapse works has 2 parameters in it for single neurons. It might be the time constant of a neuron, basically how quickly it responds to its input, and the resting membrane potential, basically what's the bias at which that neuron is sitting. And we have no measurements for this, so we have to figure out how to get that measurement. And similarly, for synapses, we know that 2 neurons have, let's say, 5 synapses between them. How strong is one of them? So that's another biophysical parameter that we don't know. Now, the advantage we have with the connectome is that we basically know the weights of this network because that's something the connectome tells us. The disadvantage is that we don't know basically the transfer functions of the neurons. Now, this is the complement, the exact complement to the standard deep neural network training, where we fix the activation functions and we're learning the weights. So this should be easier because there's only n of these parameters as opposed to n squared potential weights. There's only n of these. But actually, we can do much better than that. Because neurons come in cell types, and cell types tend to make the same kinds of proteins, that we can come up with a massive reduction. So we can make this approximation that whatever those biophysical properties are, they should be the same for all neurons with the same synapses. So here's that approximation that we made, which brings us, for a network of about 45,000 neurons, just 700 parameters that we have to estimate. If we didn't have the connectome, then it would be, really on the order of millions of parameters that we'd have to fit. So massive reduction in number of parameters, but still we have to figure out how to fit those. So here, the hypothesis that we made is whatever those parameters are, they should help the fly see the world. And so let's pretend that this is a deep neural network and then train those parameters such that this model, this simulation, can see the world. And in particular, the fly is really good at detecting motion. So can we train this simulation to be able to successfully detect movement in the visual world? And then ask, once we've done that, whether we can, the neural activity that results matches real neural activity. So, so this is what we did. In neuroscience, we call it task optimization. So, it was trained to kind of match an optic flow prediction task, which is where you have video clips that are shown, and this network, which is a system of 45,000 couple of differential equations, needs to be able to detect movement from one frame to the next, do this sort of temporal processing that compares one frame to the next frame and figures out how each pixel moves from one frame to the next. We were able to train this model. We did this, and then we asked, once we brought all these ingredients together, how good is our model? The nice thing about the fly is that from animal to animal, the nervous system is highly conserved. Basically, when you think about nature versus nurture and how the brain looks, it's largely nature. So genes have a big effect. And so the connectomes are quite similar from animal to animal. So that means even though this brain for which we have the connectome, that fly died over 10 years ago, we can take measurements that people made in other flies and ask, how well does the neural activity from one fly match the model that I made in another fly? And the nervous system is so conserved that these neurons have names, and people can look for the same neuron from one animal to the other. So what we did was we curated about 26 studies that had made recordings in various neurons within the fly visual system. And we asked, for these neurons, can we predict how the measurements made in the lab? So one of the biggest discoveries in fly visual neuroscience a little over 10 years ago was the discovery of neurons called T4 neurons and T5 neurons as what are called the elementary motion detector neurons. So these are neurons that are the first neurons through this network that are motion selective. They come in 4 flavors each, and some are prefer movement in leftward movement. Others prefer rightward movement, up and down. And so they're sensitive to and selective to movement in these 4 cardinal directions. And these come in these 2 flavors for bright edges and dark edges. So we asked, imagine that you were teleported to a world where you first had this connectome before you were able to record neural activity. In order to discover these neurons, people had to just kind of hunt around and look at different, record from different neurons and check and see whether they were motion detectors or not. Now, in this alternate reality where we first had the connectome and we used our model to make the prediction of which neurons would be motion selective, how good would our predictions have been? And in this case, perfectly. Because we would have predicted that these would be the elementary motion detectors, which is super exciting. And in fact, if you look across the 64 cell types that we have, we show that these which are supposed to be emission selective, are. Their inputs are not, and they should not be. And similarly, people have only, out of these 64 cell types, which have been really some of the most studied, it's still only been a small fraction of these that people have ever been able to record from. And there's many, many more that have never, ever been recorded from. So the question is, can our model make predictions for those? And yes, we can make predictions for neurons that you have never, ever, nobody's ever recorded from before, which is super exciting. And across a variety of different measures that we looked at, summarizing these 26 studies, we basically got them all right, including what's known as the segregation into on and off channels. And we have predictions for all of these uncharacterized neurons. And that's something that you could never do without this sort of model. I'll kind of skip. This is really a mechanistic simulation, so you really have predictions at the level of single neurons and the voltage dynamics that each neuron has. So you can make predictions of things that you could never measure. So I'll kind of skip over all. So what we did here is we trained a neural network to perform this prediction of optic flow. Now, if I had done that with a black box neural network, we would never be able to make any of these predictions. It's only because we constrained this model with the true connectome that now we're able to look at the hidden units inside this network. And because they have a one-to-one correspondence with the real brain, now make these predictions at this level. And so this shows, A, the power of the connectome in making the hidden units identifiable, and B, the power of these AI methods to fill in parameters that you could never measure directly. So that's super exciting. So can we extend this from this, this model that we've built of the optic lobe to the rest of the brain, for which we now have the connectome? Well, the thing that we needed was the connectome, and we needed the inputs and the outputs of that nervous system in order to be able to do this task optimization. Now, at the scale of the entire nervous system, what are the inputs and outputs? The inputs are the sensory system, the output is the motor system. And so, the inputs, these sensory system and motor system, they come from the body. The nervous system is embodied within a body, and the body is what gives it its inputs and outputs, and what tells the nervous system about the environment around the animal and its interactions. So what we did was we built a physics simulation of the body and the environment in order to embody the nervous system. And so what we did was we imaged the body in very high detail. This is something my colleagues at Janelia are really good at, the anatomy. And then built a 3D model of the anatomy, and then brought that into, in collaboration with folks at DeepMind, a physics engine, which we then allows us to build a physics simulation of how the body moves as opposed to just an animation. Now, once you have this physics simulation, you can imagine using the same kinds of reinforcement learning techniques to get it to behave, walk and fly. And by RL standards, it worked perfectly, but by biology standards, it failed completely because when you train it to walk, it walks completely unrealistically and flies completely unrealistically. So the thing that we did then was to try to constrain this with data. So we went to used data from our colleagues down the hall who recorded videos of flies walking around, and then used that to then train this fly to walk. And similarly, we used data from Michael Dickinson's lab here at Caltech where they recorded flies flying and then trained this model to fly. Now, I should note that the fluid simulation here for the flight is extremely simple. It's a really embarrassing to be in this audience and talk about it. And so I'd love to chat about maybe how we can improve the quality, the realism of the fluid side of things. But now we have a simulation that we think is quite realistic, but as well comprehensive. It has all the body parts, it has all the degrees of freedom, which we can now use to embody at entire nervous system scale. And so what can you do with something like this? So you can virtualize an experiment. So here's a male courting a female. It's a little hard to see, I apologize. We can, let's see if I can play this again. Now we can virtualize it. And so here are virtual flies that are behaving in exactly the same way as the real fly. And we can then predict exactly what the sensory experience of each of those animals is, both its visual experience as well as other sensory signals. And similarly, what are the motor outputs of this system, what they have to have been in order for the fly to move in the way that it did in the lab. And so this is information, these are predictions that we can make with this body model, which we can then use to build our model of the nervous system. And so that's sort of my dream. So I'll end with a vision for what I think is possible. So this fly, this male fly that has 166,691 neurons, that is smaller than AlexNet. That's a much smaller network and has fewer parameters, given that we've already measured the connectome. In the way that we're trying to go about this, it has far fewer parameters than, and we have so many measurements we can use to constrain this. We have, the connectome. We also have transcriptomes, which can inform these biophysics, not directly, but maybe indirectly. And we can collect lots of behavior. So how do we bring all these pieces together along with neural activity measurements that have been made? So these are measurements across scales and across decades as well of time and labs. But this can serve this model as a way of integrating these measurements to build, for the very first time, a simulation of the entire nervous system at single neuron resolution, where each and every one of those neurons and synapses corresponds to a real neuron and synapse in the brain. This would give us a way to go from, here's a fly performing a behavior, here's how the neural network underlying it works, how it does the things that it does, the mechanism behind thought. So with that, I'll take questions.
Great presentation, Srini. Quick question. So the starting point, and you drove it really well, was the connectomics and the existence of these big data sets. So I wonder, as a Gedanken experiment, let's assume if you had the activity of all neurons, but were lacking the connectome, how would you be able to go about it in the same framework?
No, and this has been shown too. There have been, both in simulation as well in other cases, having the neural activity alone doesn't constrain the connectivity. There was a very large number of connections, mechanisms. So it's basically, you're asking the question of predicting causality from observational data, and it's not, it's fraught. So that's sort of the challenge. Whereas if you have the connectome, that tells you what causality is possible.
Yeah, great talk, very inspiring. I was curious, when you got to the body and the flying and walking, I missed when you're doing the RL. Is it still connected to the kind of brain model, or
Right
you're
now,
just
there's
directly?
no brain there yet.
Okay, so what is it? So you have, but you have a.
You have an artificial neural network.
Okay, but you have an idea of like where the. I mean, you have controllers for essentially the different muscles or something, and then later on you try to connect those. Yeah. Thanks.
Exactly right.
And is it conscious? Is your simulated fly conscious? Yeah.
Is it real fly conscious?
So it seems like part of the reason that you can sort of envision doing this is that all flies have essentially the same brain, and I guess that's because they don't live very long and they'd better figure out how to do things fast. Well, I mean, humans then take the years to train their brains, and we all think kind of differently, which is a good thing. How much of this is going to transfer well?
Yeah, so right now we have no way of getting a connectome for a whole human. These microscopy techniques that we're using just don't scale. So I'll just put that, but let's put that aside. Let's pretend that we had it. Right now we're in this world where it's kind of like we had the genome for the first time. There's genes and proteins inside of there. We don't know their function yet. Once we start to know them, then we can say, well, oh, if there's this neuron and that makes these ion channels inside of it and has these proteins, well, then maybe we don't have to train those parameters anymore, infer them through this process. We just know what they might be. And so there might be fewer parameters then, which we can potentially, I'm just going to do a thought experiment here. So let's say somebody's recorded your entire behavioral experience. Maybe that sort of information could be used, sort of constrain the parameters that are still unknown. But I'm reaching a lot here. In practice, sort of the next big connectome project that's underway right now is a larval zebrafish, which has a similar number of neurons, but it's a vertebrate. So that's exciting. The nice thing about the larval zebrafish is that it's transparent. So people have used microscopes to image neural activity across the brain. And so this animal whose connectome will hopefully be released in the next year or so, we have both its neural activity while it was still alive, as well as its connectivity, once we complete that. And so that will be really useful as a code book.
Amazing.
Great. Well, that brings us to the end of our morning session. Again, and thanking our speakers.